So thank you everybody. Today we're gonna do the lecture number 37 and 38. There are only 2 lectures more to finish the module of endocrinology. And the part 1 of this module will focus on male and female reproductive endocrinology. So what are the learning outcomes for this lecture? By the end of this session, you should be able to define the structure and functional components of the hypothalamic pituitary testicular axis, discuss the endocrine control, the endocrine and paracrine control or the Leydig cells and the Sertoli cells, which are very important in the spermatogenesis or the formation of the sperm. Define the structural and functional components of the hypothalamic pituitary ovarian axis. Discuss the endocrine interaction that give rise to coordinated ovarian uterine cycles. This will be in the second part of the lectures. And describe the menstrual cycle in relation of the hormonal and physiological changes. That will be explained in the second part of the lectures on Sunday, I believe. So just to recapitulate and give you a lecture outline, we're going to cover the gross anatomy of the male reproductive system. After the anatomy of the male reproductive system, we will explain how this is regulated at the level of the endocrine level through the hypothalamo-pituitary testicular axis. We will study the spermatogenesis, the main testosterone biosynthesis, and functions of male hormones. Then we will move into the gross anatomy of the female reproductive system, the HP ovarian axis, and then we will study contraception and menstrual cycle in the other part, because today there is a lot to learn. So if we go into the gross anatomy of the male reproductive system, so obviously the male reproductive system is in the pelvic anatomy, and we have here a cross section, sagittal section. And if we start, the main reproductive organ that obviously produces the endocrine are the testes, which lies here outside of the abdomen, and the testes are surrounded by the epididymis and connected with the vas deferens, and they are inside of the scrotum outside of the body, because the temperature is very important to be few degrees below of the normal body temperature. Now this, which the main function is to produce the sperm, is connected through the vas deferens to other type of organs, such as obviously the penis, which is composed of the urethral opening, where the obviously here we can see the glands of the penis, the foreskin, the shaft, the ductus deferens is connected to the testicles. But importantly, what you can see is that the testicles are connected with a gland here, which is called the prostate gland. The prostate gland obviously produces secretions where the sperm is motile, and it's contacted through here, obviously, to be released. There are other structures such as the bladder before, right? And also the urogenital diaphragm here, corpus cavernosum, and corpus spongiosum. So those are the main gross anatomy of the male reproductive. So if we look through another type of vision, what we can see is that there are the testes. Obviously the testes are important because are the most, not only they produce the sperm, which is important in the fertilizing the egg, but also they produce hormones that virilize the fetus, and basically this hormone is testosterone. So we have here the testes above the epididymis, where the sperm will be kept before the ejaculation, and the vas deferens, and then a series of glands which will produce liquid that helps the motility of the sperm, such as the seminal vesicles here, prostate gland, very important, and the Cowper's gland here in just before the release through the penis. So what about the anatomy of the testes? The testes are a pair of glands, and there are two, maybe outside of the abdomen in the scrotum, appear firstly in the abdomen early in life, and then they descend via the inguinal canal into the scrotum towards the end of the fetal development. When there is abnormalities in fetal development of the testes due to, for example, infertility because there is not enough gonadotrophin, there is obviously the effect of that the testicles do not descend, right? Non-descending of the testicles may be a sign of infertility, and obviously it appears because they are not in the scrotum as cryptorchidism. So the testes, if we look inside of each testis, are composed of seminiferous tubules. Seminiferous tubules contain two types of very important cells, Sertoli cells and maturing sperm cells. The Sertoli cells are very important in the production of the sperm and in the spermatogenesis. They are packed in between the seminiferous tubules, and what we can find packed in between are the Leydig cells, which are also very important in the production of hormones. If we look at the anatomy of the testes, we can see here a sagittal section, right? So this is the position, so it's a sagittal section of the testes. And what we can see is that we have the testes here, which basically it's separated into lobules, separated by a septum, and they live inside the seminiferous tubules. You can see there are lots of seminiferous tubules. Seminiferous means that will produce a sperm, and these tubules are separated by septums to form lobules, and they are inside of the testicle. They are inside of the testicle, separated by the tunica albuginea and tunica vaginalis. So this is the main testicle, and the sperm is produced here, and the sperm travels to the epididymis, which is this structure here. And the epididymis can be divided into head of the epididymis, which contains also these tubules, body of the epididymis, and tail of the epididymis. as you can see, is attached to the testicle through this tubulus rectus, or this a connection to the tubules. It is also vascularized by blood vessels and a spermatic cord. If we look at the epididymis here, it can be divided into five parts anatomically. 1 to 3, which is called caput epididymis, and 4, which is corpus epididymis, and 5, cauda epididymis. Here we have seen the gross anatomy of the testes, but what we can see here is another cross-section of more or less the same, okay? What we can see here is that we have the testes with the septum, and here are the tubules, and they are connected to the epididymis, head of the epididymis, tail of the epididymis, through these efferent ductules, okay? So it connects, and the sperm will travel to here into the head of the epididymis. Then this epididymis is connected to the rest of the reproductive organ by the vas or ductus deferens, okay? Here we can see again the cross-section is exactly the same as we've seen, but I wanted to show a little bit more a different way of showing it. The testis, each specific globule that contains seminiferous tubules, each one, and is separated from one another. And then we have here the connections, and then this is the epididymis, head of the epididymis, body of the epididymis, tail of the epididymis, contacting the rest through the ductus and vas deferens. So if we look again into the gross anatomy, we could separate each part of the testicle, of the septum, containing each lobule, right, with Lewin cells, okay? So remember, there is the testicle, epididymis, vas deferens. But if we concentrate in this region here and amplify it, we go to B, okay? And what we find is a very classical structure which is formed between the basal membrane. You can see here the basal membrane. Then there are Leydig cells around it, here, the basal membrane, and then the Sertoli cells. And within the Sertoli cells, the developing spermatocytes in yellow, that all connect to a lumen of seminiferous tubules where the sperm will be released. So if we look at the sequence of what is happening in here, is the production of the sperm. Now, if we look in this slide, you can see the basement membrane. Now the basement membrane is here. So what you can think of this is a cross section of round. This goes in a round, okay? So we have the basement membrane, the spermatogonium, which will be the precursor of the sperm, okay? And these will be surrounded by Sertoli cells. One Sertoli cell here, another Sertoli cell there. So what we can see, right, is that the spermatogonium here, okay, is surrounded by basal membrane component, and then it separates and forms the primary spermatocyte. The primary spermatocyte matures into a secondary spermatocyte, and the secondary spermatocyte then matures into a spermatid, and then forms the future spermatozoon, which has the cap and the tail. Once this spermatozoon has finished its differentiation, it's released into the lumen of the seminiferous tubule. The Sertoli cell produces hormones which are important for the sperm maturation. And as you can see here, they have mitochondria and also the nucleus of the Sertoli cell. So if we look into the histology of each testicle, what we can see is the process of meiosis. So it's basically starting from a diploid cell into an haploid cell. But you have studied that in cell biology, I believe. So we can see here a cartoon of a cross section and the histology here. And what we can see at the top, right, is the spermatogonium. So these cells here are the spermatogonium. Then they will start differentiating and go into the nucleus, and we can see here the tails of this spermatid, right? So again, there are the interstitial cells, capillaries, and then we have the spermatozoa and the dividing spermatocytes. And you can see here very clear this structure and the spermatids and spermatozoa in the middle, right, with its tails, ready to be released because they are mature. So we're gonna see a video recapitulating this information on the general anatomy. Please do pay attention to the video. See if it works. Why is this not being? 1 second. Maybe it's uploading. 1 second. I don't know why. Sorry guys, we're gonna
so
start.
This is a tutorial on male reproductive anatomy.
Now, oh, there is a problem with this.
Okay, so this is a tutorial on male reproductive anatomy. So I'm going to talk...
I think I will have to show it like this.
Okay, so this is a tutorial on male reproductive anatomy. So I'm going to talk you through some of the main structures relating to the male reproductive system. And we're going to begin with the testes, which are the primary sex organs of the males. So they're akin to the ovaries in the females. And the main functions of the testes are to produce sperm and to produce male hormones, so androgens, and the main one being testosterone. So in utero, the testes actually descend from the abdominal cavity via the inguinal canal and they enter into the scrotum. So what we're going to do now is I'm going to switch over to a diagram of the testes, and we'll look at it in section and we'll talk through some of the key structures to know about. So the testes are actually surrounded by a capsule. So this is formed by a layer of peritoneum which is brought down with the testis when it descends from the abdomen. But this connection between the abdomen and the testis eventually gets broken, and it leaves the testis surrounded by its own separate capsule. So you can see that I've drawn on in green this outer capsule, and this is called the tunica vaginalis, and it has a parietal layer and a visceral layer. And then in blue you can see that I've drawn on another layer. So this is the inner sac which encapsulates the testis, and this is called the tunica albuginea. And as you can see here, the tunica albuginea actually forms these projections which project into the testis and divide it up into lobules. So these projections from the tunica albuginea are known as septa, and the space that you have between each of these septa are called lobules. And then within each of these lobules you've got this coiled tubular structure, so you've got the seminiferous tubules. So at the end of these coiled seminiferous tubules, you've actually got a straight tubule coming off. So you can see these straight tubules, which I'm highlighting in yellow. And these straight tubules then all join together to meet this network of tubules, which is known as the rete testis. So in Latin, the word rete means net. So you've got this sort of amalgamation of tubules forming this net-like structure. And then next in this series of tubules, coming off the rete testis, you've got the efferent ductules. And the efferent ductules connect to the epididymis. So posteriorly on the testis, you've got the epididymis, which you can see that I've outlined in purple. So what happens essentially is that you get these sperm cells which are secreted from the lining of the seminiferous tubules, and then they travel via this duct system which I've just described to be stored in the epididymis. And it's in this location which they're stored until ejaculation. So just coming back to the 3D model, you can see the epididymis here sitting on the posterior and superior aspect of the testis, and it's got three parts. It's got the head, it's got the body, and it's got a tail inferiorly.
Okay, that was a little video to recapitulate the anatomy of the reproductive male system. So now we're going to, obviously now that we know that the function of the testis not only is the fertility and reproduction, but also the main production of a sperm, but also the production of androgens and the hormones important for virilization and also lots of other function. So, and the main hormone is testosterone, right? So, and this is regulated by the hypothalamic pituitary testicular axis. And if you remember on the lectures of the first week, we studied the hypothalamus pituitary, and we will recover this and recapitulate. So, we're going to study the hypothalamic pituitary testicular axis, and what you can see here is the hypothalamic and the pituitary gland, and basically GnRH, or gonadotrophin releasing hormone, through the portal system secretes gonadotrophin releasing hormones and acts on the pituitary gland to express luteinizing hormone and follicle stimulated hormone, which in turn activates in the mature testicles the production of testosterone and also inhibin and activin. These hormones have at the same time a negative loop feedback into the GnRH, so they basically stop GnRH production, and also when there are high levels of testosterone, also into the production of LH in FSH in the pituitary. At the same time, the lack of the pituitary hormones produces also a negative feedback in the GnRH. So this gets a little bit more complicated because there are other hormones that we're going to study. So if we start in the hypothalamus pituitary testicular axis, we have the LH stimulates the testosterone. And where does the testosterone start? In the Leydig cells, right? Remember the Leydig cells, which are the ones that are around the um, formation. Let me just show it to you again, okay? Not to get confused with the Sertoli cells. So these one are the Leydig cells, okay? And the ones that are in contact with the sperm are the Sertoli cells. So LH stimulates the production of testosterone in the Leydig cells. FSH stimulates Sertoli cells to support spermatogenesis and synthesis and secretes growth factors such as inhibin. Testosterone exerts negative feedback at the same time to stop the production of LH and FSH. Inhibin exerts the feedback to stop the production of FSH only. So here we have the diagram. This diagram is like a bit more complex of what we had, but basically it summarizes the hypothalamus, secretion of gonadotrophin, the secretion in the pituitary gland, the main target organ, which is the testes, and the production of the testosterone and the positive and negative feedback loop. Normally they are all testosterone, and which is the main androgen, and in a negative feedback loop it inhibits secretion of in the hypothalamus and anterior pituitary. This is exactly the same diagram but a little bit more understandable, and what you can see is the expression of testosterone from the anterior pituitary gland. The testosterone from the testes, right? The basically has a strong inhibitory effect in the pituitary gland to secrete FSH and LH, but also it has inhibitory effect in the GnRH production under the hypothalamic involvement. Then also testosterone, we will see that can be converted in estradiol in peripheral tissues, but we're not going to touch these now, okay? So you can see it has this feedback loop negative effect in the anterior and the hypothalamic hormones. So if we start from the hypothalamus, so from the way up here, okay, from the GnRH, we did study this at the beginning of the lectures, but we're going to go over again. So the hypothalamic drive is the release of gonadotrophin releasing hormone. Okay, gonadotrophin releasing hormone is secreted in a pulsatile rhythm manner. And it's produced by the parvocellular GnRH neurons and have intrinsic pulse generation. Remember that the gonadotrophin releasing hormone neurons do not appear, they are not born in the hypothalamus, but they migrate from the nasal placoid into the hindbrain, into the brain, and then they set in the median eminence, where they project axons, and these axons will be in contact with the portal system, and these ones will release the gonadotrophin releasing hormone that will contact the pituitary gland. If there is a problem in the formation of these neurons, either at the level of secretion or at the level of migration, they produce Kalman syndrome or infertility because there is no GnRH from the hypothalamus. So the GnRH is transported in the hypophyseal portal circulation. Remember, I think it was lecture number 5 and 6. So you can also go back to this part. And remember that basically what it does once it reaches the pituitary, it activates the GnRH receptor, and it stimulates the expression of alpha GSU and LH, luteinizing hormone, and follicle stimulating hormone. This happens via a G-coupled receptor, activation of cyclic CMP, PKA, and CREB. And it stimulates the secretion of LH and FSH of the gonadotroph cell type in the anterior pituitary. And obviously, gonadotrophin releasing hormone receptor is not prone to densitization or down regulation, and that was also explained in lecture 5 and 8, so you can have a look at that. The important bit is to understand that GnRH regulates anterior pituitary secretion of hormones of LH and FSH. So how about the hormonal control of the testes from the pituitary? So the anterior pituitary secretes gonadotrophins from the gonadotrophic cells. And these ones are two main hormones. One is the luteinizing hormone, and the second is the follicle stimulated hormone. And they are both heterodimeric glycoproteins, and they have a common gonadotrophic subunit precursor gene, which is alpha GSU, and that will specifically translate into LH beta and FSH beta subunit. And so basically there is different genes, LH and FSH, from one common gene, alpha GSU. So the hormone products of the testes are Leydig cells, which synthesize testosterone. The Sertoli cells synthesize and secrete inhibin and also androgen binding protein ABP. Remember, there are two different types of cells. The Leydig cells are next to the tubules, whereas the Sertoli cells are the ones that are intimate contact with the spermatids to form the spermatozoa and to form the sperm. Both of them produce paracrine factors, particular members of the TGF-beta family, such as inhibin, which suppresses FSH, and activin, that increases FSH. So obviously, these ones, for example, right, the Sertoli cells produces inhibin, suppresses FSH for the production of the, there is a decompensation, the production of the sperm. So what type of hormone is inhibin and activin, and what type of the receptors do they bind to? So if we look into the cells, this is a little bit what happened, right? So we have the Leydig cells and the Sertoli cells, okay? The Leydig cells, right, have the LH receptor, so LH binds to the receptor, it activates cyclic AMP that activates pKa that goes into the nucleus, and what produces is the enzymes. Remember from the lectures 23 and 24, these enzymes convert cholesterol into testosterone through different enzymatic reactions. The testosterone produced in the Leydig cells travels through the membrane into the Sertoli cell. It can be converted, it can be converted through aromatase into estradiol, which at the same time can signal the other cell. Okay? But in the Sertoli cells, they are susceptible or responding to LH instead to FSH instead of LH. So the FSH will bind to the receptor, produces cyclic CMP, pKa, go into the nucleus and start producing the cells required or the enzymes required for the production of the spermatid and the spermatozoids. As you can see here, it produces inhibin, AVP, and also aromatase, so converts testosterone into estradiol. And this allows the formation or development of the spermatozoids. So if we look here into the different cell types, again, we have the Leydig cells that produce testosterone synthesis. And then we have the Sertoli cells, these very, very big cells, which engulf the production of spermatozoids. And they are very important in the process of spermatogenesis. The Leydig cells are responding to LH, whereas the Sertoli cells are responding to FSH. And it's the combination of both that will generate here a mechanism to the maturation of the final sperm. So we have studied now the process, the anatomy, and HP axis regulation of the male reproductive system. So now we're going to study the spermatogenesis process. So basically, what we need to understand is how from an undifferentiated precursor there is the production of spermatozoids or obviously the differentiated sperm. This starts with a cell which is undifferentiated at the basal membrane in conjunction with a Sertoli cell called spermatogonia. The spermatogonia will undergo cellular division and also duplication of their genomic DNA and will form the Sertoli cell. The spermatocyte. And the spermatocyte will mature into the spermatids. And these cells will be transformed into the final spermatozoa by adding the cap and the tail. So again, if we look at the different cross sections, you can see the dividing spermatocytes and the spermatids and the spermatozoa in the lumen of the cells. And we can see here when in the histology, H&E staining, we can see this process when it culminates. So those are a sequence of events in the formation of the final spermatozoid. So we start from a spermatogonium, and then there are different types of cell divisions, which I think that you guys have studied in cell biology, which basically you start from a diploid cell and you end up from a haploid cell. And these different steps are divided into leptoten, zygoten, pagiten, diploten, and deakinases, and will form the gametes. So then you have the secondary spermatocyte, and finally round spermatid. So those are the known process of spermatogenesis. So round spermatid then elongates a spermatid to form the spermatozoon. And the final spermatozoon looks pretty much like this. So it has a head, it has a neck, middle piece, and a tail. If we look at the head, obviously the tail is very important for motility, but if you look, has a nucleus, connected piece, proximal centriole, coarse outer layer, outer doublet of microtubules, central pair of microtubules, annulus, and then obviously we'll have the tail. This is very important for the motility, and that will be at the end the final differentiated spermatozoon. So this will complete the effects of spermatogenesis. As you can see, this is a complex mechanism whereby it requires the hormones LH and FSH in two different cells to act on the Sertoli cells to initiate two different things. One, the gametogenesis to end up from a diploid to an haploid cell, and also the morphological changes required for cell determination, cell differentiation of the final spermatozoon. So how is this regulated under the endocrine system? So complete spermatogenesis depends on FSH, testosterone, and DHT, which we have studied. FSH more important in early stages of meiosis in the Sertoli cells. Androgens are more important in later stages plus the spermatogenesis. So the epidemial maturation, the spermatozoa still needs to acquire motility and ability to fertilize the oocyte. So both acquire sperm pass through the caput and caudal epididymis. Sperms acquire decapitation factors added during the storage in caudal epididymis but removed prior to fertilization in the female tract. So there are four potential sources. Remember that testosterone is very, very, very important. So there are four, the function of the testis, apart from production of the sperm, is the production of sex androgens, which is in males very strong testosterone biosynthesis. And this occurs in the testes. And remember from the lecture 23, 24, where we studied the main factor is cholesterol, right? So the cholesterol, right, it's required for testosterone biosynthesis. And it can be synthesized de novo from acetate, from plasma membrane cholesterol, cholesterol esters or lipid droplets, or plasma lipoproteins LDL or HDL. So, major endocrine stimulus to testosterone biosynthesis is LH. Acts via luteinizing hormone chorionic gonadotrophin receptor also, and basically is coupled to a G and cycle CMP through the PKA CREP signaling. It also, and this is in lecture 23 and 24, remember, it's acute stimulation of the STAR TSDP BDAG, which is a complex of enzymes, and the CYP enzyme. Chronic upregulation of the TRD and CYP enzyme genes will give to the testosterone biogenesis. Remember that the rate determining step is the mitochondrial and uptake of cholesterol. Cholesterol within the mitochondrial oxidases to pregnenolone. Two options for downstream metabolism of pregnenolone, right? And remember, we studied this in the other lecture of the steroid biosynthesis. So cholesterol can be converted to pregnenolone, and then through the 17 alpha hydroprognenolone enzyme give rise to the DHEA and androstenedione, and androstenedione gives to testosterone. Also, we have the pregnenolone can give to progesterone through the same enzyme 17 hydroxyprogesterone can give rise to androstenedione. Okay, and those are the main mechanisms to the formation of testosterone. I'm just putting this, if you remember, from lecture number 23 and 24 of important enzymes in the production of the steroid hormones. Remember, the first one, mineralocorticoids, were in the adrenal gland. The second part, glucocorticoids, so that was in the different zones of the adrenal. And then this one is the production of glucocorticoids and androgens. As we just seen, they can have cholesterol through pregnenolone, then 17 hydroxypregnenolone to the hydroepidrosterone, DHE, and that it gets converted to androstenedione and testosterone. And also testosterone, we will see that can be converted in estradiol. You don't need to worry about the first two, the glucocorticoid and mineralocorticoid, were studied in the adrenal, right? So remember that it was in the zona glomerulosa, zona reticularis, and this was in the zona fasciculata, but the main steroid production of the hormones is also in the testicles, where this conversion can occur. So what is the function of male cells hormones, right? Testosterone is responsible for secondary axis characteristics during puberty. It increases aggression and libido, enlargement of the larynx of the voice, male pattern of pubic hair, maturation of the genitalia, muscle development, sperm production, bone growth, and acne. So with this, we're gonna finish the first part of the lecture, and then we're going to have a break, and we will study the anatomy of the female reproductive system. Okay? The anatomy and the function of the female reproduction system is more complex, as you will see, because the female anatomy and endocrine system needs to give rise to the ovulation and circadian ovulation of menstrual cycles of every month. But we will study this at the end of the lecture. We're going to have now a break. Thank you very much.